Hey listeners, this is episode 739 of People Behind the Science. I'm your host, Dr. Marie McNeely, and today I'm looking forward to speaking with our guest researcher, Dr. Beth Weaver. This episode is made possible with support from our sponsor, New England Biolabs. Listeners, are you curious how molecular biology is moving forward around the world? Hear from the scientists who are driving discovery and expanding applications of existing technologies on the New England Biolabs podcast, Lessons from Lab and Life. Host Lydia Morrison interviews revered scientists working in genetics, synthetic biology, diagnostics, molecular cloning, and other fields. Get ready to explore the cutting edge of science and learn what these remarkable scientists have to offer. Don't miss out on the opportunity to hear exclusive stories about the world of laboratory research. Tune in to Lessons from Lab and Life today, wherever you listen to podcasts. And today, Beth will be sharing stories about her life and career. So listeners, get ready to meet another one of our fantastic people behind the science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to People Behind the Science. Today, I am thrilled to be speaking with our guest scientist, Dr. Beth Weaver. So, Beth, welcome to our show today. How are you?
I'm good. How are you?
I'm doing quite well.
Thanks for having me.
Of course. And we're looking forward to learning more about you and your research, Beth. But let me first take a moment to tell our listeners a little bit about your background. So listeners, Beth is a professor in the Department of Cell and Regenerative Biology, as well as the Department of Oncology and McArdle Laboratory for Cancer Research at the University of Wisconsin-Madison School of Medicine and Public Health. And she is co-leader of the Developmental Therapeutics Program at the University of Wisconsin Carbone Cancer Center. Beth received her bachelor's degree in biochemistry from Brown University and her Ph.D. in biomedical sciences from the University of California, San Diego. Afterwards, she conducted postdoctoral research at the Ludwig Institute for Cancer Research before joining the faculty at the University of Wisconsin-Madison, where she is today. Now, Beth has received various awards and honors during her career. These have included receipt of the Bothwell Prize and the Women's Health Research Mentorship Award from UW-Madison. In addition, she has been named an American Cancer Society Research Scholar, a Romnes Faculty Fellow by the Wisconsin Alumni Research Foundation, and a University of Wisconsin Carbone Cancer Center RIDE Scholar. And Beth, today we'd like to get to know more about your science, but also more about you. So what do you like to do when you're not doing science?
Well, my kids are 14 and 11 right now, so we do a lot of things as a family. We like to do family Halloween costumes. So this year, my older daughter was Cruella de Vil, and my husband was her henchman. And my other daughter and I were Dalmatians.
That's awesome.
Yeah, and in previous years, we dressed up as Hocus Pocus characters. And then the year before that, we did Candyland. I was Fun Dip, and my daughters were Cotton Candy and Tootsie Roll. And my husband was a Sugar Daddy, which was a little on the nose.
Well, I love that. Now, does this creativity spill over into other areas of your life as well, or just Halloween costumes?
Well, actually, we really like to get together with friends and family, including entertaining. And my youngest recently turned 11, and we hosted a Harry Potter themed birthday party. And so we turned the living room into the great hall with the head table and then the 4 house tables and the floating candles. And after the kids came in and the wands chose the wizard, then we had a sorting ceremony where they fit into a cake pop and the inside color would tell them what house they were in. And then we had a series of scavenger hunts in which they would find the common rooms. We decorated various rooms in the house as common rooms, and then they found Horcruxes. So yes, we like to do lots of fun activities as a family.
Well, this sounds absolutely amazing and quite detailed. I'm very impressed. Well, Beth, it sounds like you and your family have a lot of fun outside of science. And I know you do some phenomenal work in the lab as well. And we'd love to talk about your scientific side. So how do you describe your work to someone who's outside of your field or maybe not in science at all?
Well, we study a group of commonly used chemotherapy agents, and even though they're considered highly effective, only about half of patients benefit from them. And so we're really focused on improving their use in two ways. First, we want to be able to identify a method to predict whether a given patient is going to benefit from treatment with these drugs. And second, we want to find a way to sensitize the large number of resistant tumors. And accomplishing both of those goals would be facilitated by understanding how they work. And this is something that we as a scientific community thought we knew for almost 40 years. But in collaboration with my clinical colleagues, I was able to look inside specifically breast cancers and find out that, whoops, we were wrong. It turns out that these drugs don't act the way we thought they did. And the reason for that is that, like all drugs, they have concentration-dependent effects. And so, for instance, when you have a headache, you take 2 tablets of your favorite pain reliever. You don't take the whole bottle. And in effect, we were treating ourselves in the lab with the whole bottle of drug. But now that we understand how these drugs work, that really facilitates our goals and will put us in a much better position to convert these drugs from conventional chemotherapy into personalized precision medicine.
Oh, well, I think this is absolutely phenomenal. And I look forward to digging into the details of your research during our conversation today. And I think working in this area of cancer research can be highly motivating and inspiring. But I think sometimes even on the best days, you need a little boost of motivation or inspiration. So do you have a favorite quote or a saying or something that really just keeps you going?
I think my motto is that failure is a prerequisite to success.
Mm-hmm.
And for anything complicated, I think that's really true. I also like to think of science and scientific projects as like a roller coaster that I haven't ridden before. And so when I think about it this way, I think there's always an upfront investment, right? If you're going to ride a new roller coaster that you haven't ridden before, first you have to dedicate time to be able to go do it. And then you have to buy tickets to the amusement park. And you have to figure out how you're going to get there, which often involves plane tickets or a drive or both. And then once you get there, you wait in line. And then you get to the ride and you wait in line again. So in science, there's often a lot of upfront investment and training and literature searching before you start your project. And then when you finally get up to the roller coaster, sometimes you have to put your stuff in a locker and there's this gut check moment, right? Am I really going to do this?
Right. Should I turn back?
Exactly. And I think of it like the go, no go experiment where you decide, am I really going to proceed or not? And then if you do, you get in the roller coaster and you strap yourself in. And the first thing that happens is you have to go up this steep hill, right? And you just push and push and push. And often it's pretty jerky. It seems like it's taken forever. But then when you get to the top, often there's a moment, you have this view that you've never had before. And then you're off, right? Arms in the air, huge smile on your face, and you get the rush. And then there are typically a few twists and turns. And then sometimes there's a second hill and you have to push and push and push again. But then you get another rush. And when you get off the ride, you think, oh man, I can't wait to do that again.
Oh, I love this metaphor.
I often find myself in the push, push, push times where you just have to push and push and push. And so when I think about, the rush is coming, that helps motivate me.
I think the hills can be steep sometimes in science too.
Indeed.
Well, it's not just about finding these sources of motivation and inspiration. I think part of that scientific journey is finding people that you can connect with and look up to and learn from in many cases. So do you have people that really stick out in your mind, Beth, as people who are mentors or role models or people who you really looked up to?
Well, I'll tell you, my role model right now, someone I really admire is a current colleague, Dr. Dusty Deming. He's a physician scientist, a medical oncologist, and he's also a cancer patient. He and I, along with Dr. Christian Capitini, are co-leaders of the Developmental Therapeutics Program in our cancer center. And at my first external advisory board meeting, Dusty came to that meeting literally with a portable chemotherapy drip. And about a year later, when we had our site visit for the cancer center, he came up with 3 tables that really, I think, encompass the breadth and scope of our very large developmental therapeutics program. Those tables were the talk of the site visit. And he really has a very remarkable way of seeing through all the noise and all the distractions and focusing on what's most impactful. And he really impresses me.
That is remarkable. I think, finding those people who have the clarity to really hone in on the key details and not be distracted by all the minutia is difficult.
Yeah, absolutely.
Well, it sounds like you found a great colleague there, and you're obviously very passionate about the work that you do. And I think this is an important area of research, but it's always interesting to hear about how scientists got to this point in their career where you're a professor at a university doing this dream job. So can you take us back to the beginning and tell us a little bit, Beth, about how you started in science and maybe what first sparked your interest?
I think I had a pretty unusual scientific journey. Most of the scientists I know were interested in science as a child, and I really wasn't. In fact, I can remember being about 5, and it was shortly after the Summer Olympics because I'd seen a bunch of interviews with gymnasts. And I was thinking, well, I don't know what I'm going to be when I grow up, but I could be anything but a doctor or a gymnast because all the gymnasts started training when they were 2 or 3. And at 5,
Right.
I was way too old for that. And I knew I couldn't be a doctor. At that point, I thought all doctors were physicians because when a surgery would come on TV, I couldn't watch that. I don't have the stomach for that. So I thought my very limited understanding of science, that was not for me. And by the time I made it to college, I still didn't understand that the job I have now exists. I thought that people who trained in biology were either sort of doing ecology or they were physicians. And I was initially undecided. And then when I declared a major, it was an anthropology major. It turns out that Brown has a really strong Egyptology program. And so there was a short period of time I thought I wanted to be an Egyptologist. I can remember going to the library and looking for books, and they weren't in the normal position because they were oversized. And so I'd be going to these parts of the library I'd never been to before, and it was dark and sort of lonely and kind of creepy. All the lights were on motion sensors, and, most, but not all of them would come on as you came into that part of the library. And finding these books, and they were a meter wide, and they weren't bound. They didn't open like any book I'd ever seen before. They had 4 flaps and they were tied together with a ribbon like a present. And then you'd open it up and there'd be these individual pages with drawings of grave sites and grave goods and hieroglyphics. And it was like a mystery with clues. It was really fascinating. And so I thought I wanted to be an Egyptologist for a very short period of time until I learned that you either have to be independently wealthy or very good at fundraising in order to do that. So I thought, well, that's out.
Not a salesman.
No, it's a little bit ironic considering how much fundraising is involved in being a scientist, but at least there are established mechanisms for that. So that was about towards the end of my second year of college. And I had a friend who said, you should really try out the lab. I think you're a scientist. And at that point, many Brunonians spend their junior year abroad. And I had already arranged to be at Antioch College, which is a small liberal arts school in Yellow Springs, Ohio, that operates on the co-op system where you work for 6 months a year. Because I thought, I really don't know what I want to do for my career, but I think that actually doing it full time for 6 months will give me a really good idea of whether it's a good fit or not for me. So I went to Antioch and I took some science courses and that went fine. And then I did 6 months in Joan Steit's lab at Yale. And it was a large HHMI lab. And it was really a lot like a big family, but a diverse family from all over the world. And we would have lab parties and went to her home once. And I really enjoyed being in the lab. We were doing molecular biology, cloning and DNA preps, that sort of thing. I thought it was a lot of fun and the people were fantastic. So I went back to Antioch. I took some more science classes. Then I did 3 months in Gary Rednick's lab at Yale. And by that point, it was pretty clear that I wanted to go to graduate school. So when I went back to Brown for my last year, I took an immunology course and I thought, oh yeah, I want to be an immunologist. So when I got to the University of California at San Diego, or UCSD, for graduate school, I lined up 3 rotations in immunology labs and I took immunology courses. And towards the end of my 3rd rotation, I've been learning all along about, these different immune cell types and what they do and how they mature. And there were always these suites of cytokines. And I was really trying to figure out what each of these cytokines do. And there was a class and I said, okay, so this cytokine is pro-inflammatory. And they said, no, it's anti-inflammatory. But in last class, it was pro-inflammatory. Yes. Well, it depends on the context. And I said, oh, okay. So this suite of cytokines is pro-inflammatory and that suite of cytokines is anti-inflammatory. And this one can be either depending on the context. And the answer was no, they all vary depending on the context. And at that point, I said, okay, that's it. I'm out. I don't understand how I'm ever going to figure this out.
I think that's amazing. I think that really emphasizes that, a lot of science is more complex than what you sort of learn about in the textbook in these early years of your training.
Absolutely. Absolutely. So yeah, I was a little lost. And I was also taking at that point a course that was taught by Don Cleveland on reading the primary literature and landmark papers. And he had sent me an email and asked to meet with me. We had to write a paper at the end of the class, but he thought I had the best paper and he wanted permission to share it with subsequent classes, which could have been done by email, but he asked me to come and meet him. So I did. And he asked me how things were going. So I told him. And he said, well, you could rotate in my lab. And his lab is unusual in that there are two sides to it. There's a neuroscience side and a mitosis side. And the neuroscience side involved surgery. So mitosis seemed like a better fit for me. And when I got in the lab, the more I learned about mitosis, the more questions I had. And I just got more and more fascinated with it. And so I really became hooked. And then when it was time for me to do a postdoc, certainly the expectation was that I would find another lab, but I wound up staying in the same lab. And I can't say that I recommend that for anyone else, but it was the right choice for me. And I did it because all throughout my graduate career, the rationale for studying mitosis was that errors in mitosis cause aneuploidy, an abnormal number of chromosomes, which is a hallmark of tumors. And actually, it wasn't uncommon at that point for people to write aneuploidy causes cancer, but there would never be a citation because there wasn't actually an experiment that demonstrated that. I really became fascinated with that question. I really wanted to know whether aneuploidy could cause cancer and did cause cancer. And there was a mouse model in that lab that would allow me to answer that question. And just as importantly, there was a very talented graduate student, Alon Silk, that was willing to collaborate with me to do the mouse work so that I could actually answer that question. And so that's why I stayed. And then when it was time to look for faculty positions, I have a two-body problem. My husband is also a scientist, and we did a very targeted search. This is also something I can't recommend. I think a broad search is the right approach, but we took a targeted search, and this is where we hid. And we've stayed in Wisconsin because it's such a fantastic place and the people are wonderful, and we've had great collaborations. And I'll call out specifically my collaboration with Dr. Mark Burkhardt, who's a physician scientist and a breast cancer medical oncologist. And we have joint lab meetings and journal clubs. And I think physician scientists in general are often transformative because they serve as translators between the basic Ph.D. scientists and the clinicians, and they allow us to do experiments that we just really couldn't do otherwise.
Certainly. And I know you're doing some really cool experiments these days in your lab there at UW-Madison. So can you tell us a little bit more about one of these projects that you have ongoing at the moment, Beth, that you are just the most excited about?
I'm excited about everything we're doing in lab. But there's one project we've been working on for a long time, and we just got the samples today that we've been working for 5 years to get. So I'll back up here. So the project is to develop a biomarker to predict whether a specific patient is going to benefit from these microtubule poisons. And based on about 20 years of research, what we think is that chromosomal instability can serve as this biomarker. And chromosomal instability is recurrent missegregation of chromosomes over multiple divisions. And it exists on a spectrum. So normal cells and some cancer cells don't exhibit chromosomal instability, so they have a stable number of chromosomes. Some cancer cells do exhibit chromosomal instability, and in the grand scheme of things, this is missegregation of a relatively small number of chromosomes. And if you induce that in an animal model, you see that it doesn't always, but it can increase the incidence of cancer. But we don't see very high rates of chromosomal instability in tumors. And if you induce that experimentally, you see it causes cell death and tumor suppression. And this is just because cells need a minimum number of chromosomes to survive. If you have a really abnormal division and you wind up with a cell that only has 1 or 2 chromosomes, it's not going to be cancerous, it's going to be inviable. And so there's this maximally tolerated threshold for chromosomal instability. And now that we understand that microtubule poisons are inducing chromosomal instability rather than mitotic arrest, as we had long thought, this suggests that the tumors that are closer to that maximally tolerated threshold to begin with are the ones that are sensitive to these microtubule poisons. And we were able to test this in a small cohort of metastatic breast cancer patients, and it looked really promising. So about 80% of the patients above this chromosomal instability threshold responded, and about 80% of the patients below the threshold did not respond. But it was a small study and it was retrospective. And so we've been working for about 5 years to get samples from a large prospective clinical trial. And we just got the first 2 boxes today. So I'm really excited to be able to test this hypothesis and to hopefully be able to bring this biomarker test into the clinic so that we can spare patients that aren't going to benefit the side effects that come with these treatments.
Absolutely. I think you're doing remarkable work, Beth. And as we've hinted at, the work that you're doing sort of overturned this really long-held belief in science about how this particular type of cancer drug actually works. So what was that experience like for you, kind of seeing that your results initially didn't match what was going on in the literature in the past and trying to, I guess, convince everybody that this was something that was really happening?
Yeah, I would say it's ongoing. I remember being in graduate school and treating cells with one of these microtubule poisons, and they were supposed to die, and man, those cells didn't die. And so I thought I did something wrong. And I think multiple times I've had the situation where the results I got didn't fit with the conventional wisdom. And of course, for me, at least, my first concern is that I didn't mess anything up. But once I've convinced myself, then it's a matter of trying to convince everyone else. And it's always a slow process. And particularly with the understanding of these microtubule poisons, this is what we've thought for decades. This is what I was taught as a graduate student. And so it takes time to change people's minds. And I understand that, but now we've looked at multiple microtubule poisons. We published multiple papers. We're building on this. And I got an email this week from a colleague that I really respect saying that our work is really transforming our understanding of how these anti-microtubule drugs work. And that is really gratifying. And I hope we continue to change more minds because I think it's really important that we understand how these drugs work, particularly since they often serve as a model for new drug development. And if we understand how they actually work, we're going to have much more success moving forward.
Definitely. And I think this is an amazing story, Beth, that, as you mentioned, is continuing to evolve as you build on these findings. And listeners, if you're looking for more exciting stories about science, New England Biolab's podcast, Lessons from Lab and Life, is your source for staying informed about the most up-to-date molecular biology breakthroughs. Host Lydia Morrison interviews prominent scientists and delves into their research to uncover how their findings affect society in meaningful ways. Tune in to learn from the experts and get a glimpse of the cutting-edge advancements being made by scientists around the globe. Don't miss out on this opportunity to gain new knowledge and stay informed on the latest discoveries. Tune in to Lessons from Lab and Life today, wherever you listen to podcasts. And Beth, we touched on some of the challenges that you've faced in your career, particularly in these situations where you're turning over just some of the long-held dogma in the field. And I think it is so important to take the time to talk about the challenges in science, because the stories that most people hear are these rare success stories. So do you have another example of a major challenge or a failure or perhaps a struggle that you've encountered in your science?
Yeah, so there have been many struggles, I would say.
Mm-hmm.
And I'll give you 2 answers to this question. The first one is a few years ago, I really couldn't get editors to send my papers out for review. There were a couple of topics I just couldn't get funded. It went on long enough that I was really starting to get depressed and reconsider what I was doing. And I thought, I've overcome all of these challenges. I thought eventually this is supposed to get easier, and it really wasn't. So I thought about the roller coaster, right? I thought about the hill at the beginning, and you got to push and push and push and push. And there will eventually be a time when you get the downhill rush. And my department chair said, oh, well, the grant I just got funded was on the fourth submission, and that made me feel better. So I think the challenges don't end. However, I recently saw this TED Talk by Ori Alan where he talked about the struggles in science. And now I think that this should be required viewing for every graduate student, everyone in science. So he talked about when he was in graduate school, he started out and he was working on a project, and it seemed like the experiments weren't working and the experiments weren't working and the experiments weren't working. And his basic assumptions didn't seem to hold true. And he stopped shaving. He had difficulty getting out of bed. But eventually, he kept trying and he figured it out. And then he published a paper that made it seem like it was a direct line from the question to the answer. And then he started a second project, and the same thing happened. When he eventually got over it, he asked his other lab mates, and they said, oh yeah, the same thing happened to us, but we never talk about it because in science, we write papers, and it seems like a very linear process between the question and the answer. But actually, it turns out that the answer is often not what you thought it was. And he described this place in the middle between where you start with what you know, that's point A, and then you think the answer is point B. And on your way there, you get misdirected. And he called that the cloud. And it serves as the boundary between the known and the unknown. And so now his students can come to him and say, Ori, I'm in the cloud. And he can say, oh, you must be miserable.
Right.
And this means that you are in the transition stage between the known and the unknown. And so he can step up support for them so that they can be more creative and find their way out of the cloud. And so I think that's really helpful to understand that everybody goes through this. And when the answer doesn't turn out to be what you expected, the process of finding out what the answer actually is confusing and problematic. And you have to try new strategies and keep being creative. And so there are going to be many times when you face this, but when you get to the other side, it really feels like an accomplishment.
Absolutely. I love this story. And I love kind of normalizing and making sure people are aware that this is part of the process and it's not just them, right?
Yeah, absolutely.
Oh, we don't just want to talk about the difficult times. We appreciate you sharing your experiences, but we'd love to hear about some of your successes as well, Beth. So do you have a favorite success story, whether it was a big win or maybe just a small but particularly meaningful victory?
Well, I think we had a nice paper come out last month, and I won a mentoring award earlier this week. And I already mentioned this email I got from a colleague talking about how our view of how these drugs work is changing. And I find that to be a real success. But I think it's also important to celebrate the small successes. And I'll give you an example of that. So we've been trying to label a particular protein for some time, and we'd gone through 3 or 4 different antibodies that didn't work. And recently, my student came to my office and he said, come to the microscope. And I came to the microscope and it was beautiful. It was beautiful staining. And so I think it's important to celebrate those small successes as well.
Absolutely. I think anytime a conversation starts with dragging you to the lab, you know it's going to be good.
Yes.
Well, that is amazing. And you mentioned this mentoring award that we also mentioned at the top of our interview here, and I'd love to talk about this as well. Can you share what this means to you and how important mentoring is in your career?
Well, I think mentoring is one of the best parts of the job, right? Watching students take ownership of their project and really start imagining how the processes work. There's really a transformation that they undergo, and being part of that process is really gratifying. And I will say that those times when they come and take me to the microscope, I live for that. Or they come in with the data and we talk about it, and sometimes it supports the hypothesis and sometimes it doesn't support the hypothesis. And then we think about, well, goodness, like what else could be happening? That's really so much fun. I get texts from my students that are really enjoyable. One of them has been collaborating with Dr. Rich Hellberg's lab, and they're doing colonoscopies in mice. So I will get texts at various times of day and various places of mouse colons. And I'm always so excited, and I can feel her excitement. So it's really worthwhile.
That is amazing. And I think these unexpected results and exciting projects can really suck you in. And science can be a little bit all encompassing. So we try to encourage our listeners to take a step back, to maybe think outside of their particular area of expertise by reading broadly. So do you have a recommendation of a book that you've enjoyed, whether it's related to science or not?
I do, actually. And I will admit that I have had more time for audiobooks than regular books. So this is a book I listened to. It's called The Power of Fun by Catherine Price. And generally, I think in our society, we consider fun for adults to be frivolous and the sort of thing you do after all of your responsibilities have been met. And I can't remember the last time I met all of my responsibilities. And so it's so undervalued that we've even cheapened the word. We'll often say that anything we do that's not for work is considered for fun. And that involves things that are really not fun, in addition to the things that you come out of thinking, that was so fun. When can we do that again? And the hypothesis in this book is that the antidote for the 3-hour faculty meetings and the endless trainings and the long line of rejections is actually having those moments that are so fun, what she called true fun. And so she came up with a fun squad of about 1,500 people around the world and asked them to send in examples of what they considered events that were so fun, so that she could come up with a definition of so-called true fun. And she found that it is the confluence of 3 things: playfulness, you can't care too much about the outcome of what you're doing; connection, which can be to nature or to a pet, but is usually towards a person or other people; and flow, which in this case is being so engaged in what you're doing that you lose track of time. And that when these 3 things come together, it results in true fun. And that true fun is really restorative and rejuvenating. And I think she's right. And so I've been trying to test this hypothesis.
I like that you're taking the scientific approach to it as well. Well, this is an awesome recommendation, Beth. We will put this out on our website for our listeners to find The Power of Fun. And one of the fun things about being in science, in addition to these amazing projects you get to work on and phenomenal people you get to work with, are the places that you get to travel. I think this is something that's often undersold in careers in science, but you do get to go to a lot of places to work with collaborators, to go to meetings, etc. So Beth, do you have a favorite place that your science has taken you?
Well, the first international meeting I had the opportunity to attend was in Cortona, Italy. And it was such a beautiful place. And the food was so amazing. And this was the first place that I learned that the house wine is the best. And it had a beautiful view at the top of the hill. And I can't wait to go back.
Oh, it sounds amazing. In your first international meeting, it's a difficult experience to top, perhaps.
Well, I have to say that I have enjoyed pretty much everywhere I've been. And I was in Portugal more recently, and I can't wait to go back there either.
Well, it sounds remarkable. If our listeners are able to either visit Cortona, Italy, or to go to Portugal, do you have particular places that you went to, or maybe specific things that you saw or did that they must put on their itinerary?
Cascais, Portugal. It's on the water. It is spectacular.
Well, it sounds amazing. Listeners, we'll definitely have to put this on our list of places to go. And throughout our conversation, we've talked about some of the phenomenal people that you've worked with over the years, Beth. And I think these people make science so remarkable. So we love talking about these quirky personalities and just the fun stories that you share with your colleagues, because I think these go against the stereotypes that a lot of people still have about scientists, unfortunately. So do you have an example from your own lab of a quirky tradition that you do, or perhaps from a past lab, or just some funny or fun memories with colleagues that you can tell us about that go against these stereotypes and show this human side of science?
Well, the equipment in my lab has fun names. So our microscopes are Shira and Adora. They're based on the Shira characters, and all our refrigerators and freezers have different Muppet names. But we also have a lab tradition of making art, and art that's related to mitosis because it is so visually stunning. So one of my former students made a series of black and white images of stages of mitosis. Another one gave me a pillow in the shape of a chromosome with a banding pattern on it.
Oh, wow.
And a current student just made a multipolar mitotic spindle out of different colors of wire.
Oh, that is amazing. So where do you put all of these cool mitosis art pieces?
Well, some of them are in my office and some of them are in their office space so that we can all enjoy them.
Oh, that's amazing. And I love that what you do is very visual. So do you ever consider, having like lab Christmas cards or something where you take pictures from the microscope, which are gorgeous in some cases, like you said, and using them for different purposes.
That's a great idea.
It's about that time of the year, right?
Yes, it is.
Well, Beth, I love to hear that your lab has this creative side as well. So do you participate in the making of mitotic art?
I have not yet. So far, I just enjoy it.
Maybe a future Halloween costume. You could work something out.
Actually, my favorite lab Halloween costume is where they dressed up as the mitotic spindle and the mitotic checkpoint. That was fantastic. I often show that at the end of my talks because it is my favorite lab picture.
That is amazing. And I think having this creative, engaged, fun group to work with can really help you when you're solving big questions and dealing with big problems in science. So if we took away some of the problems that normally hold you back, Beth, and we took away the barriers like funding, staff, technology, feasibility, not a problem, what is the one research question that you would want to answer most?
Oh, man, I'd really like to have two.
Mm-hmm.
Well, one is I'd really like to be able to tackle mitosis in every different cancer type. Many of the therapies that are available or coming online will target specific pathways that are functional in a subset of tumors, but all of them have to go through mitosis. And so there's a common vulnerability there. And I'd love to be able to target that in every different cancer context. The other thing that really appeals to me is being able to determine which precancerous lesions are going to transform into full-blown cancers. For instance, in breast cancer, mammograms have identified lots of ductal carcinoma in situ, and most of those would never progress and become a problem in the woman's life, but there's no way to predict that right now. And so there's lots of overtreatment. And so I'd love to be able to find a way to determine which of those lesions really need to be treated and which of them are never going to pose a problem.
Interesting. Well, this is fascinating. And I love these questions and just thinking about the impact that they could have both in kind of broadening our understanding of mitosis and all these different types of cancer and kind of digging into this sort of precision medicine issue of understanding or predicting who's going to develop a cancer and who is not in these cases to sort of personalize treatment a little bit better and not just, like you said earlier, throw the whole pill bottle at it in every case.
Right.
Well, thank you for dreaming big with us on the show today. And I'd love to end our conversation, Beth, by talking about advice. So for our listeners out there who might be on their own scientific journeys or other adventures, do you have a piece of advice that really helped you that you can share with all of them today?
Well, my graduate advisor, Don Cleveland, always used to say, aim high, because even if you miss it, you'll wind up somewhere worth being.
I love it. Well, is there any other last piece of motivation or inspiration that you would like to share with listeners?
It's all about what you want. Keep trying.
Wonderful note to end on, Beth. If our listeners want to learn more about you and the remarkable work that you're doing, what is the best way for them to do so?
Just search for Beth Weaver Lab in any browser.
Fantastic. Well, listeners, definitely check out what Beth is working on, learn a little bit more about her lab and the phenomenal research they're doing. And Beth, it's been such a pleasure to have you here with us today. We truly appreciate your time.
Thank you so much for having me.
Well, it was great to chat with you. And listeners, wonderful to have you here with us as well. We hope you'll join us again for our next episode of People Behind the Science.